And then, well, in parallel, we decided to go to different strategies, not only gene therapy strategies, but also some pharmacological strategies also. We tried this, remember the early 797. And yeah, that's what we have right now, and I can go further later.
Hi everyone, so my name is Dan Moore. I'm a postdoctoral fellow in the lab of Severio Tedesco. So I'm based in London, in University College London, in the Francis Crick Institute. So I finished my PhD last year looking at developing stem cell-based models of laminopathies. So we build 3D engineered constructs in the lab that replicate muscle tissue. And so the hope is that by using human cells and a more sort of physiologically relevant environment, we can more accurately mimic patient phenotypes in our lab models. And so what we want to do is to use those models to be able to start to generate new gene therapy approaches, but also look at some more basic disease mechanisms that aren't possible to do in other model systems like mice, for example, because of the way the genetics works is slightly different. So that's kind of broadly what I do, and yeah, we can talk about that.
Oh,
Okay.
Hi, Mark Corman. My son is almost 6 years old now. He's 6 at the end of the month. He has the R249W mutation, the whatever most common one. Yeah, I don't know. That's about it.
Hi, I'm JP Gorley. My sister is 7. Her name is Kate, and she has the R249W also. Yeah.
I am Shannon O'Callaghan. I have a 12-year-old. Madison Manning is my daughter. I don't know her mutation. I know that it's on paper, but it's not the most common one.
Hey, this is Göknur Haliloğlu. I'm a pediatric neurologist in Ankara, Turkey. I'm mostly dealing with pediatric rare disease, but within that content, I'm just seeing neuromuscular patients. A great privilege and a pleasure to be here with you on this meeting. Yeah.
Okay, so I think the best thing would be to expand a little bit more about the 3 projects that, well, 2 specifically on LMNA, and Rotem can also maybe mention about the outcome measures.
Should I start? Well, the projects that we are involved in, as I was mentioning before, so mainly are focusing to...
Sorry, I think... Hello. It will be good to have one mic there and one here,
yeah.
And so we can go back and forth with questions. It doesn't need to be...
Yes, I was saying before, so we are building new cellular models, so we are taking cells from patients, and we are working both with myoblasts, and now we started with induced iPS cells that can be used in different experiments, and we can get differentiated cells from there. But probably the most interesting tool for us was the mouse model. We developed our R249W mouse model that we have used for 2 things, mainly to better understand the underlying mechanisms of the disease, and then to use it as a platform in order to test different therapeutic strategies. Regarding the therapeutic strategies, we decided to use, in this case, CRISPR-based strategies, because in this situation, with a dominant negative mutation, we believe that is the most interesting one. And we started what we thought at the beginning that was actually was the only thing, so CRISPR 1.0. So at that moment, that complex was only able to destroy the DNA. So that's we decided to destroy the mutation specifically, and to keep the wild-type allele intact. So that was our first strategy. The second one was also based in that system. It was called HITI. It's called HITI, and we can integrate it. We can integrate a wild-type allele in front of the mutant one. And the third one that we are trying to develop right now is one based in CRISPR 2.0. It's a base editor. In this case, we can edit exactly the mutation and change the mutation to the normal one. And these are the 3 strategies. We have results in cells for all of them, and we have also the validation of the 2 first strategies in the mouse model. And now we are trying to finish the experiments for the validation of the base editing. In this long trip, as I was saying before, this 10 years, We were accumulating different results, and this is not a straightforward history at the end, because the mouse model, for example, doesn't recapitulate the disease completely. So we have the cardiac phenotype in the mouse model, but we don't have the skeletal muscle phenotype there. We don't know why, but that's what we have right now. And we were able to obtain animals that are homozygous for the mutation, something that doesn't happen with the patients. But they gave us also important information about the energetic and metabolic phenotype that we were talking before. And we are using that also to better understand the disease. And we have different phenotypes that the therapeutic strategies are not working equally in all these scenarios. And this is giving us, again, information that is not always the information that you want to have, because you want a complete solution, but so far that's what we have.
Did you say that it worked for the cardiac,
Yeah.
but not the muscular, or did you say it worked for the muscular but not the cardiac?
So there are two things here. So first, the animal model recapitulates the disease, just talking about the cardiac phenotype, okay? That's the animal. And regarding the therapeutic strategies, there is only one working for the cardiac phenotype. That's what we have right now. That is the one that consists in destroying the mutant allele. And this is more the most important project that we have.
Is there any other animal model that recapitulates the skeletal phenotype?
As far as I know, with LMNA, there are no animals. I know that Mark is also developing the same animal that we have, but with the humanized exon 4, in order to avoid one of the steps of validation and just using always the same guides. They are going to be the human ones. And as far as I know, the animal is equal to the one that we have, no skeletal muscle phenotype. And this is a very interesting result, but we don't know, because we can use that in a therapeutic way in the future, if we know why these animals are not recapitulating the skeletal muscle, but I have no idea about how can we...
Isn't that fairly universal across muscular dystrophies, though? That the mouse models don't recapitulate?
No, not at all. In other muscular dystrophies, you get yeah. Actually, we were prepared in order to do all the experiments in order to get the phenotype, the skeletal muscle phenotype, but we are doing the experiments, but no differences so far. There are some experiment that is missing, that is the regeneration of the muscle to induce an injury or something like that, and to see if the regeneration is fine or not. But even though this is going to give us a phenotype, we should have an original phenotype, muscular atrophy there, but we don't have it. And this is it.
So I don't know, maybe I can speak a bit more about my PhD project and what I did there. So I was using induced pluripotent stem cells primarily, so I wasn't using the mouse model. So these are human cells that have been treated with certain factors to make them more like an embryo. And so what we can do is we can grow those in the lab, and then we can do a process called differentiation, whereby we make the tissue that we want to make in the dish in the lab. And so the advantage of that is that we can start to look at these diseases in human cells, not in mouse cells. And so, for example, we don't need to have a homozygous mutation to elicit a phenotype. And so what we want to do is to make a sort of a screening platform, so whereby we can use, for example, these CRISPR tools on these cells in the dish and see how effective they are at recovering normal sort of healthy cellular phenotypes. So what I did was we had we had cells from 3 different patients. One was a K32del mutation, one was the R249W mutation, and then we also had a very rare mutation that resided in a specific part of the gene. And so my PhD work firstly focused on just making sure that we could see the sort of characteristic changes that we observe in patient cells, in patient muscle cells, in our model in the dish. And so what we were looking at in the context of laminopathies was changes in nuclear shape. So we were able to see that in our model. And so what we did was we applied similar CRISPR tools to these cells. So we focused on this rare mutation, this one in exon 5. And so the advantage there was that that has been proposed that a potential exon skipping strategy, so where you can actually fully remove part of the gene rather than targeting a mutation very specifically, that might have the advantage of being able to target various different mutations that all reside sort of within that neighborhood within the gene. So we carried out two strategies: specific correction of this mutation and also this exon skipping approach. And what we found at the end was that really specific correction using these CRISPR tools worked much better. So the exon skipping, for some reason that we haven't investigated fully yet, doesn't seem to fully restore kind of healthy phenotypes to these cells in the dish. And so the advantage of this approach is that if we encounter another rare mutation, we can use these cells in a preclinical model before putting any potential therapy into a patient. So that's kind of the broad approach that we took. One thing that we also sort of saw within the study, and I think I should maybe highlight, is that one thing we were looking for was whether the cells with the LMNA mutation were able to make muscle as efficiently as cells without the LMNA mutation. And so what we did was we did this process of differentiation, and we didn't see any difference in the LMNA mutation cells versus the wild-type healthy cells. And so what we think sort of comes out that's actually different from cellular models where you take a muscle cell directly from a patient, and they do have a sort of reduction in the amount of muscle tissue they can make. And so I think what that maybe points to is that if we, develop these CRISPR therapies or whatever sort of therapeutic approach we do take, that early intervention where possible is probably going to be better, because we think that sort of acquired damage to the cell across the lifetime of the individual sort of compounds and makes it more difficult for the cell to do its job properly. So yeah, we use the kind of basic CRISPR tools that Ignacio was talking about, but we are starting to look at using the more advanced, more modern technologies that are coming, and we're looking at trying to package those into what are called AAVs, so viral vectors that will be used eventually to deliver these CRISPR tools into patients. And so the idea in the future will be that we can take these CRISPR tools in these viruses, put them onto our model system in the lab and see how effective they are. And that will enable us to screen different strategies before going into a patient potentially with very rare mutation. So we used some of the most common mutations, the R249W, but we also wanted to look at these super rare mutations, because I think LMNA particularly has lots of different variants that can cause lots of different phenotypes. And so, yeah, this is kind of the approach we took to answer some of these questions.
Did you treat the cells and then differentiate into myotubes to see the correction, or did you treat the myotubes or the 3D muscle and see a correction of the actual thing that's already formed?
Yeah, so where we're at the moment is we treated the stem cells first and then differentiated into the muscle cells. So where we want to be at is where we can differentiate the muscle cells and then apply these viral vectors. So that's what we're working on at the moment. But so far, we want to show first that the CRISPR correction was able to rescue these phenotypes just before they've become muscle cells, to make sure that works. But that we now know works. So the next step for us is to really begin to use these models with those viral vectors, exactly as you said, and to treat the muscle cells in the dish directly.
And is there any, outside of the nuclear morphology, have you guys found any functional phenotypes, or have you been doing anything with functional phenotypes, whether it's force or fatigue or anything like that in these cells yet?
So that's, sorry, go on.
Or just for now sticking to the morphological changes, I guess?
So that's work that we're doing at the moment. So what we want to do is essentially we put these cells in what we call a hydrogel scaffold. So if you can imagine, you have two posts, and then between them we have this artificial muscle that's sort of free hanging. And so what we want to do when we electrically stimulate the muscles is we want to see them pull. We want to see them do what a normal muscle does. So what you'll see in the post is this contraction of the post due to the action of the artificial muscle construct. And so ideally, what we would do is we would take a direct measurement of the amount of force that the muscle was able to apply to these posts, and then we can use that to say, post-treatment with the CRISPR, is more force being produced by the muscle compared to the cells just with the LMNA mutation that are untreated. So that's technically it's quite challenging to do that, but it's what we're working on at the moment. So our lab doesn't just work on laminopathies, we work on various different muscular dystrophies, collagen 6, central nuclear myopathy, and that's kind of a strategy that we're trying to apply to essentially all of the diseases that we work on. So it's very much work in progress. We're not there yet. There are kind of quite a few technical challenges in regards to the cell culture and how you measure that force effectively, but I think it's something we'll get to. And we do have some promising early results, not in laminopathies, but in another muscular dystrophy. So I think we're getting towards that. The other thing I can say is we also look at the transcriptome. So this is if you have a gene that makes a product called RNA, and that RNA is what is used sort of as the blueprint to make a protein. And so what we want to do is to look at globally within the cells is the types of RNA that are being produced in the LMNA cells different to the control cells. So we started to do that work as well, and what we're kind of starting to see is that the cells almost have their own inflammatory profile. And so that's something we'll be really interested to see after correction. Can we restore a non-inflammatory transcriptional sort of state within the cells, and those experiments are ongoing as well. So we're trying to look at morphological, transcriptomic, functional, and really kind of have an array within the model of outputs that we can assess.
And have you seen from the transcriptome area, do the mutations all have the same inflammatory signature, or is it different between different mutations?
It's a good question. So we're looking at them as a group, so laminopathy versus non-laminopathy. I have the data back now. So the technical challenge there is that if you have cells from different people, their genetic background is going to be very different, and so it makes analysis of those transcriptomes slightly tricky. What we have now, where we have done the CRISPR correction on these cells, they're now on the same genetic background, so you can much more easily compare those cells and the transcriptomes of those cells. So we have just received that data back. I haven't analyzed it yet, but I think it'd be really interesting to look at what are the transcriptional differences between the mutations and whether there are different pathways that can be targeted between, for example, the R249W mutation and the K32del mutation. So I think that'd be really interesting to do, and we're working on that.
It's very relevant question in terms of using anti-inflammatory medications, especially in early onset, a bit more severe phenotypes, including steroids as well, to selected populations. So within the continuum of the spectrum, knowing that, is it a primary thing or is it something secondary that will just really help us to just certify our treatments on clinical grounds as well?
I don't think I can say for sure yet. The early transcriptional data we have looks like there is some sort of primary inflammatory signaling coming from the muscle cells themselves, but how that would relate to secondary inflammation after muscle breakdown, what's the balance there? I think it's too early to say in our study yet, but it'd be something that's really interesting to look at. And so kind of one idea we have is to take the cells that we have in the dish and put anti-inflammatories on those and perform the same transcriptomic analysis and see if that might help the cells even without sort of intervention at the genetic level. But yeah, it's a really interesting point, but we still need to work on that.
I'd like to add something there. This anti-inflammatory and these glucocorticoid treatments and all those things is something that I always heard, that you should do this and blah, blah, blah. And even when there is not a clear inflammation phenotype in the patient, it looks like they worked. And I keep asking why, and nobody tell me why they are working. So we started a project in order to try to understand more or less these things. So right now we know that the glucocorticoid pathway at least in the cells coming from patients, is altered. So this is something that, as far as I know, it was not known, but now we have those data, and probably in the next years we are going to be able to try to figure out better or to get new targets that could be used in a therapeutic way instead of using the corticoids, for example. Maybe we are going to be able to think about gene therapies, applying all these guys and trying to avoid the side effects that could have with these corticoids. But this is a very interesting point from my point of view. Yeah.
I can share a little bit about my research. Hold on, let's do that. Well, my part really comes down the line, right? Like we have the scientists to work and find the therapies, but then once you really want to do the clinical translation and bring it to the patients, there are like outstanding questions that if you start thinking of them at the time that the drug is ready, it's way too late. So I think it's really good for the field, for this group and community to think of all those questions even together. And I'm focusing on muscle imaging outcome measures, but it's true also for other parts like physiotherapy assessments and other assessments and patient reported outcomes and all of that. But I'll expand on more of my expertise in what I bring to the table. So at the NIH, we're trying now to launch a natural history study for congenital muscular dystrophies. And we don't define the subtype, so all congenital muscular dystrophies could come and meet with us if you'd like to come and have like annual assessments. And in parallel to that natural history protocol, there's gonna be my protocol that deals with optoacoustic muscular imaging. So this is an investigational device that creates it really looks like an ultrasound. So you all probably know how ultrasound looks like and how the images looks like. But this time it uses light in the near infrared spectrum. So it's a laser light that you kind of fire to the muscle, and then it gives you back the optoacoustic profile. So this profile try to give you how much fat, how much collagen is in the muscle instead of healthy muscle. So if you will, it's kind of a non-invasive way to look at muscles on how much fibrosis is in there. And it's a pretty cool device. It's investigational, right? So if you go to a regular hospital, you're not gonna see something like that. It uses a laser, so we all need to wear like protective goggles because the laser if it hits the eyes, it can damage the retina. But the purpose of it is really to create an outcome measure, a biomarker, imaging biomarker that is objective, that you can apply and not put the patient in the MRI machine for 2 hours, because there was, maybe you are aware of other muscle imaging stuff that's been going in the field, and it's never easy for our patients to be in the MRI device for so long. And then also on top of that, every kid that is under 7 years old is not likely to love the noise and the setting. So many times you really actually need to sedate them to get good quality images. So this investigational device is just bedside device like a regular ultrasound, but with some of those protective stuff. And then instead of doing muscle biopsy, the outcome measure is how much fat, how much collagen you have in the tissue. So in order to really make it a valid outcome measure for clinical trial readiness, it would be really important to apply that on different muscular dystrophies. So I'm very grateful and honored to be supported by the American Neuromuscular Foundation. They are giving me a grant to do it for LAMA2 and collagen 6. And I am planning to bring also LMNA patients for the same purpose, just so we can look across different subtypes and try to actually say for the first time whether this technology is suitable or not. I think it's going to be a very strong outcome measure if we prove it's working. Of course, to prove something works, you need to show that over time it captured the progression. It can capture maybe even like the reverse, like the improvement, and that it's very kind of, that if you repeat it, it's valid, right? So like there is correlation, and if I do the exam and then Gochner comes and do the same thing, like we're gonna get the same results, that it's valid enough. So there is a lot of process into it, but that's definitely the right timing to work on that while they are preparing the drugs, because once you really have the drug ready and validated, you want to have those outcome measures ready for your clinical trial, because now FDA also is asking for those more objective kind of key biomarkers that can facilitate approval. So that's my research focus, and I really hope that I can invite the community to join us soon enough at the NIH to help us understand that. I'm wondering from my side, I want to ask you, do you think like people would like to come and to contribute their time and energy for something like that, or yes?
I think people would. How have you done it? Or you said, so you haven't done it on these muscular dystrophies yet. Has it been used for Duchenne, or has it been used for normal people / older people? Like, how have you validated that you think it might work?
That's really an excellent question. So at the NIH, we bought this fancy device, very expensive one. It's been sitting around for quite some time because we had to put in place lots of regulatory stuff, right? So like, we're not working on cells, we're not working on mice, we want to bring it to patients, and actually pediatric patients. So we had to go through all the FDA, it's IDE regulated study. FDA needs to give us special authorization to test this device on humans. We have it now in place, and right now the NIH IRB is reviewing our protocol to make sure that that everything is really in place, the statistical plan, and how we even compensate people who comes on site and decide to contribute their valuable time for research. So there is all of those components that sometimes are more challenging when you do clinical translation. So we don't have any experience yet because we are not allowed yet to put this probe on a patient, but we're definitely heading there. And there is a very big group in Germany that did that on Duchenne muscular dystrophy, on Pompe, and on normal, like healthy subjects. What optoacoustics can give you in terms of the readouts, it's not just fat and collagen, it's actually also about the oxygenation, how much oxygen is in the tissue. And now if you think of that, you can even challenge the tissue. Like you can ask someone to exercise and you can see how much oxygen the muscle was using when you did the exercise. So there could be more potential for this, and people have been exploring that. All of that are what we call proof of concept studies. So the Duchenne study was on 10 patients. There is also a study on SMA patient with this muscle imaging, and that was also 10 patients and 10 controls every time. So it seemed to be very promising, but there is still work there, and that's the reason it's still considered investigational. Otherwise, this company was already selling that as a clinical, device. Yeah. That's another great question. So we have few core muscles that we chose based on some technical consideration and without diving too deep. One is the biceps, the gastroc, the vastus lateralis here in the thigh. And the reason is because from my experience on myself, those are easy to get in kind of a good way. And then I actually listed in the protocol many muscles around, I think I have maybe 10 muscles that I want to allow myself to look at as an exploratory stuff, right? So for one example would be the diaphragm. So it's very hard to look at this muscle, but I'm not sure if it's gonna be successful. The transducer is a bit different than the one you have in a normal ultrasound machine. So, but I want to try that because if I can say how much fibrosis or how bad the diaphragm look like, it's gonna be great. So we're gonna try all of that, but the master protocol is really about those 3 muscles that are more easily captured, and then we're gonna go from there.
Always very interesting to learn these, outcome measures. I was wondering, will that require a subtype-specific readjustment in terms of, distribution of axial muscles as well? So I understand that depending on the subtype, you have these muscles groups selected, and you will pick up at the end which muscles to follow up with.
Yes,
right?
yes, that's exactly it. So like I want to do those 3 key muscles on everyone just to get a sense of how much even the device is reliable, right? Because that's part of the question for investigational device. Actually, for this kind of investigational device, you really want to know if it's safe enough. We know it's safe. Like we've been talking to investigators from all around the world now, and so it's also being utilized in the cancer, oncology field, because you can use that for other stuff. So like there are lots of applications for this device, but specifically, so we know it's safe, and we also, the FDA think it's safe. It gave us the authorization to work with the device on humans and even pediatric patients. But still, we need to show that on a larger scale. And to your question, yes, 3 muscles across everyone. Like if you get to come to be in the study, we want to know the details of that. But then, of course, for maybe LAMA2 or collagen 6 or LMNA, they're going to be more specifics into it. And we will understand it as we go. Yeah.
Do you have specific questions for any of the projects, or some questions about research in general, animal models, cell models? Or the other thing, you have a good experience of doing or trying to get research going. Do you want to give us some of your experience of the approaches that you were trying to do and still trying to do? I don't want to put you in the spot, but
I'm happy to discuss the things that we're doing if people are interested in that. Yeah, so our foundation also is trying to do some research stuff and has worked with Ignacio a lot, or I at least speak to Ignacio a lot, so I appreciate that. But sort of the approaches we've tried to take is, I guess, if you think about the different, there's kind of different levels, and I think most of them are ending up at this point on some sort of gene therapy or CRISPR type thing. Seems like you need to get to the genetic root, and if you're above that, there's so many things that go wrong. You can sort of help one thing, but it's not enough to have a big impact, is sort of what I think we found so far. So I think the things that are left that I think are most promising are the CRISPR. We're right now focusing more on the prime editing standpoint, which is, I don't know, what is that, CRISPR 2.5, CRISPR 3.0? It's CRISPR 3.0, which would be trying to just, like for the R249W mutation, trying to change that W back to an R and do nothing else. And having some progress there, but everything takes time. I think the next step up is trying to work at the RNA level, and as based on a lot of Ignacio's work, where if you destroy the mutant allele at the DNA level, it improves the phenotype. So we're trying to see if we can target the mutant allele at the RNA level and just degrade the RNA without actually destroying any DNA, because that might be a little bit safer, and if something goes wrong, you can stop taking that. And we've had some progress there as well, but I think with all these things, prime editing, ASO is how do you deliver it efficiently to muscle so that things work well. And then like the third approach we're taking is a more universal, I guess, approach to the different mutations, that it doesn't need to be mutation specific, where we're trying to silence the whole endogenous gene. So silence the whole gene and then have via AEV have something come in and replace the whole gene. And that has also, there's progress, and it seems like we have, right? It seems like we have stuff that does what we want all those three things to do in a cell culture and whatever, in the best conditions possible. The questions are, even if that does work, when you actually get it into a real model, is that actually going to give you any recovery, / is something going to go wrong in between going from a cell culture to an animal model or a engineered muscle tissue. So that's kind of the main things that we're left at, I think.
And what I didn't know that you were working on a mouse model with the humanized gene that through Jackson, or
Yes, so that is basically one of our collaborators. So I'm loosely working on it. One of our collaborators was able to get Jackson to do that under one of their grants that existed. So we were sort of the impetus for it starting, but I haven't really been, the other person is sort of managing the process. And now recently, I think Ignacio is involved with that as well, because I think they're at the point now where they're starting to get closer to trying to characterize it. So obviously, his experience with the other mouse model will make their life easier, and him having access to their mouse model will make his life easier for testing some of his therapy. So hopefully they're now seem to be working together, and we're kind of still not really involved, but that's fine, because the work got done.
So it's done. That's good. Yeah.
Yes.
okay. Any other specific questions? What should we?
The question that I have is the question you don't have the answer to, right? But it is the question. How long are we likely to wait to see this being used with our children? Are we likely to see this being used with our children?
I will keep an eye on Duchenne always, because
I see it popping up. I'm starting to see, there's this for SMA, there's this for we're starting to see that things are happening.
yeah. So yeah, we are now working on different strategies, and we are trying. So as I see, the thing is, you have to first try to select in cell models, in mouse models, which is going to be the best strategy. We are trying to do that with many strategies, because we have tested, and we are testing 3. You are testing another 3. There are other groups working in different strategies. You were also working in different strategies. So of course, if you are testing many strategies, you are going to go a little bit slow, and that's one of the things that happen. But once we have the best one, we have to wait to the really, really difficult part, that is the delivery. As far as, for me, that's the most difficult, the bottleneck right now. And this is something for all the muscular dystrophies. This is not LMNA related. It's all the muscular dystrophies. So you had to treat this disease in vivo. So you had to go there. You had to go to the cell, to the tissue, and you had to fix the tissue.
In every single cell?
Or a percentage. Maybe it's 50%. 50% is enough. And we are assuming that the tissue, the target, is the differentiated muscle, which I'm not 100% sure about that. So maybe we are talking about progenitors, the ones that need to be fixed. But let's assume that is the differentiated one. So you had to go there and you had to fix them. In another diseases that right now are being cured with these techniques, you extract the cells because it's an hematopoietic disease. So you can extract the blood cells, you can fix that in the lab, you can expand them, and you can put them back. And this is much easier, okay? But for the muscular diseases, neurological diseases, so you have to go there. your medicine has to go there. And that's why I was thinking that probably Duchenne is the one in which they are trying many things. There are a lot of efforts there. So we had to keep an eye on them, and because the delivery is going to be the issue. In the meantime, we had to work in the best strategy for us. And then the platforms are going to be great, as we were commenting. So because once you have the platform, it's easy. If you compare with the situation that we have right now, it's much easier to change something, very small thing, and try to go there. So, no answer, but...
I agree with everything you said. I would say the one thing also additionally, you can just, if you get to the platform, you can just change that small part at the end. But I think the NIH's natural history study, if they had 130 kids, I'm trying to remember the number, there was, I don't know, 78 different mutations. So you have to change that end piece a lot of times to actually hit all the kids, which isn't going to be cheap. Even though you can do it, and it will get a lot cheaper, it's going to take time for that changing end piece and really the regulatory hurdles of after you change the end piece, what they allow you to do. I think, to me, I'm probably a little bit more from the idea that if you want to look at the bottleneck to get the first kid with LCMD cured, it's probably the muscle delivery, but to get it widely, it's probably the regulatory bottleneck of changing that. How easy is it to change the last piece? Unless something changes from that regulatory perspective.
That's something that I think is going to happen.
Okay.
Because at the end, if you have the solution there, if you have the delivery fixed, it's going to be so hard to say now this is just an administrative thing. Or even if you think about the money, that is another issue. That now we are thinking about these very expensive therapies. I'm sure this is something that is also going to change. And it's going to be, you know that there are gene therapies that you can get in these cities and places with a very low cost. And this is going to change, that's for sure.
I think one thing on that is when talking about changing that end piece, and maybe you guys can comment a bit more in terms of, I keep hearing sort of the FDA is sort of beginning to push more towards organoid models rather than animal work, and so that's kind of what we're working on in our lab. And so kind of the hope is that if we can change those end pieces and show that they work as we expect them to work in our cell system in a comparatively faster manner, that might sort of speed up the process of targeting all of the different mutations that we need to target eventually. So I think there is room there and there's scope there to speed up that process by using different model systems. So yeah, it's just something I could add.
Yeah, and also the platform, they are talking, I think, mostly about the CRISPR situation. But if it's, I mean, there are some other research going on the more classical gene replacement that is difficult in LMNA because of the haploinsufficiency, etc. But there are some, for example, in other dominant mutations that people having some good results doing knocking down and replace. So using a different technique to get rid of the mutant allele and make a modification in the normal allele to avoid being downregulated because of the other tool. So and inject both genes together. They want to downregulate the bad gene and the one that it will replace the good one. Again, it's a difficult step, but there is people working on that one, and that will not need to be mutation specific because you can just get rid of the whole allele. So there are different strategies. it's good that we are advancing in models of the disease. The humanized mouse is a very good idea. Most of the field is trying to move to the humanized mouse models, so that you have a humanized mouse model means that you are putting a gene in the mouse. So you are creating the mouse model with a gene that has the human parts. So because it's slightly different compared to the mouse, the original one. So any tool that you create for gene therapy, for example, will work better, and you will not need to change it when you go into humans. So you can use it in the animal model, and you can use it in the human cell models, and then go to clinic. But yeah, it's and the other good thing about talking about platforms that the FDA now started they have a mechanism to approve platform situations for companies. So if for example, there are, in the case of Sarepta, that unfortunately we have this, I don't know if you heard about Sarepta. Okay, Sarepta is a company that had the gene therapy for Duchenne muscular dystrophy approved last year for 3 to 4 years old, and they extended to other ages. And recently they have 2 deaths to patients that had liver failure, and they were the one granted with the first platform from the FDA. So the platform means that they were using a vector to introduce the gene therapy, and so if they go to a different disease, knowing that that vector works, they can just use the other gene, but the same vector. Now we stop because of these problems, but it's good that from the regulatory side, there is also a recognition that we need to move as a platform and not one disease at a time, because that will be much more time consuming, of course.
Yeah, and I want to echo Gustavo and say that I know it can be, I don't have an answer for you. It could be in a year or 2, or, never. But I am hopeful, and I do want to encourage you that with the explosion in genetic-based therapies, every lesson learned is gonna also help the LMNA community. So whether it's a death on the Sarepta or in other programs, because there are also many outstanding questions about immune toxicities and stuff that we need to address as clinicians after we dose those patients. And those could be lessons that we learn across programs. And I do want to encourage you that we're not wasting our time. We're learning as we go from all programs. And that's why it's also very important to move to the kind of platform notion, but very, very carefully, because one thing that can be valid and applicable for one disease, for example, a disease that doesn't affect the heart muscle, could not be maybe extrapolated for LMNA. So we need to think very carefully, but as a field, we are very much trying to focus on learning the lessons together across programs. And specifically, I'm part of the ENMC meeting for AAV talks that we're gonna have in September to try to understand more of AAV. AAV is the vector we use to deliver the machinery that corrects the gene or the gene replacement. And there are some immune toxicities related to that, and to understand it better. So when finally we have maybe AAV that brings a CRISPR machinery to correct the LMNA gene, we know to treat better the toxicities and predictable adverse events. So hopefully there is hope for us as a field to move to that goal together.
Well, one of the strategies that was commented here was trying to get that kind of a strategy in order to to be able to fix all the mutations, but which means that you had to remove the expression of all the lamin that you have, both the wild type and the mutant one, and introduce an exogenous wild type one. This is something that one of the groups is trying to test, and this is included, and this is gonna, if this strategy works, it's gonna be good for all the mutations, for all of them. If you ask me, I don't know if this is gonna be the best, the strategy that is gonna work the first, because it has a lot of tricky points that you had to take into account, and I don't know if right now the technology is ready to achieve that. But we are working on that. There is a group that is working on that, of course, because as you were saying, this is the perfect, if this strategy works, it's perfect, because it's gonna work for all the mutations. Being said that, we are prepared in order to, as we were saying, to do the whole thing in a personalized way. So, but you have to start with one of the mutations. It doesn't make sense, at least to me, to work in all of them, because once you fix one, the strategy is gonna be 90% similar for the rest of the mutations, and it's gonna require these small changes. And actually the platforms right now have been created for that, in order to go one after the other and the other and the other and the other. And there is an issue here that is always on the table, that is the variable time. Time is very, very important. And it depends, of course, for you, the time goes really, really slow. I understand completely that. But for me, that I've been in this field from a scientific point of view, it is amazing. We are going really, really fast. This change, so to me, when I was studying biology, to be able to change the nucleotides, and this is something that we are doing right now, it was impossible. And now we can do that. Not only that, we are applying those medicines to the patients. Few patients in the world, but we are doing it. CRISPR technology was demonstrated to be a good technology in 2012. And a couple of patients have been already treated. So I think that we are really in a very, very fast race. And I'm sure that the techniques are gonna, imagine that in a month or something like that, there is a new technique in order to deliver all the components there. And we don't know, but it could happen. So, and there are many groups working in this, not only for the muscular diseases, for other rare diseases. 80% or 70% of the rare diseases are genetic. So it's a common interest for many, many people in the world. But I understand that for you, this is really, really slow. But there is no other way, and as far as I know. And I have a comment regarding these Duchenne death cases and all those things. And I think that is very important to keep the perspective as general as you can, because I was working in the cancer research field a long time ago, and things there are completely different. Okay, so chemotherapy is a really, really dangerous medicine, but we are used to that. I don't know why in this field we want to have a perfect medicine. So, but this is quite difficult to have it. Even cancer, that we are really used to that, we are used to see things that now we are seeing in these clinical trials. So I'm not saying that this is perfect, but it is important not to create huge expectations with these new gene therapies, because at the end they are going to have the same problems. And actually I think that they have fewer problems that some drugs like the chemotherapy, for example. And I think it's important to say these things to the patients and also to the scientific colleagues, because sometimes we are talking about things that I never heard in the cancer research field. So that's amazing.
Oh, I just would like to share that your all, elegant questions are shared by all of the communities. I mean, I do serve overseas, and I just receive the very same questions from my patients. So that I just really appreciate just this effort is really, really very important. And maybe we can just recognize that this CMD and CureCMD international registry really helps to understand and figure out, you know, the variance and distribution of variance among the subtypes, which is really an extremely important tool to be there so that we can just understand for which variant we have how many patients are out there. So even my patients are registered, you know, to this international registry held by the CureCMD. So that's just something really extremely important for the scientific community, for the scientists and also for the clinicians to follow up the pathway in terms of evolution of the disease. From the LMNA perspective, one problem is evolution of the disease in terms of not only weakness, but also the contractures, the cardiac phenotype really differs from one patient to another. So I, as a clinician, for instance, wonder if there are some other modifying factors which really changes the flow and evolution of the contractures. On the second hand, nowadays everyone is in good hands doing rehabilitation, care, which really changes the course of the disease itself. So these are all, you know, questions in my top of my head, and also this therapeutic window. And I believe in my heart that every and each community member may be, you know, included in some of the clinical trials from different aspects. Maybe not from a muscle, you know, weakness perspective, but from if you are just going to rescue the cardiac phenotype, then age shouldn't matter, to my understanding. So these are just very little questions, both in terms of therapeutic windows and these treatments and the registries in general.
And going back to the cardiac, do you have some more idea than me about the potential clinical trial that the group in
Singapore group?
the what?
Singapore group?
Yes, the Singapore group is going to do.
No, we were in it.
Yeah. There was not much information.
No much
there.
information about that.
But yeah, there is a company in Singapore that they were working in another protein that is connect with LMNA, with the lamine protein, that is also a nuclear complex called link, and they are trying to do also kind of a platform where modifying that link could potentially fix not only LMNA but also some other nuclear issues, proteins that are in the nuclear envelope, and they are preparing a clinical trial for LMNA mutations. But I went to their website to see, but there is not much information, but only saying that they have almost ready for the regulatory side to go ahead and start recruiting patients. But I don't know, you have anything else? No.
I mean, the only thing that I had heard, but I don't know if it's actually true, is that at least for a long time would never touch any of our kids, because I thought it was all like adults, dilated cardiomyopathy only that would be considered for that trial. But
Yeah, for that trial, yes.
Yeah.
But
So it's good to see them doing, I agree that doing something in the disease and having a trial where there's some is great, but for the perspective of,
no, and
yeah.
also in a conservative view, that it's probably better that they go to the adult population instead of going to your kid to do that first. So yeah, so, I mean, I bring it that because you mentioned if in your lifetime, in the lifetime of your kid will be something like this, and there is one clinical trial cooking right now for the cardiac aspect for LMNA adults, but it's closer.
I have a comment. Maybe I think that now I can tell about these things. And you remember actually this, I was commenting before about a drug that is called RE797. I don't know if you know about this one. Okay. So this drug was also, they were thinking at the beginning, it was a small company, the one that got that drug, and they decided to use, for scientific reasons, to use in these adult cases of LMNA-related cardiomyopathies. And I remember that from many organizations, they were asking me to test that drug in our mouse model and blah, blah. So we decided to do it, we did it in cells, in the cell models. We had a fly also, we did it in the fly model, and we did it in the mouse model. In all the models, it was working very well. Well, at least with our biomarkers. In parallel, Pfizer bought the company, this small company, and Pfizer decided to proceed and go to a phase 3 clinical trial. So we contact Pfizer in order to use that and to get money in order to do better experiments, blah, 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 blah. And they would say, okay, yes, we'll do it, but no agreement. So the majority of the experiments were okay, and we were doing the best experiment. That was the experiment with the animals that are quite similar to the patients. So it was really, really a bad result for us. So, and I present those data in different conferences. We didn't publish that yet, but we saw that the result was not good at all. And in parallel, Pfizer decided so that the product is not anymore in the clinical trial, is retried, so no more product at all. The reasons were that it was not good enough or whatever. So I remember many organizations asking me to push because they wanted the kids to be there in the clinical trial. So sometimes to go too fast maybe is not a good idea. You had to go to all the steps in order to know. And maybe that was a mouse issue, and it was just related with our mouse model, blah, blah, but at the end, the product is not in the market, okay? That's something that is for sure. And regarding this other strategy, that gene therapy, I'm not sure if this is going to work either. At least in the results that I saw in the mouse model were not so. And I was asking for what happened if you treat the adult animals, and you remember that they were not able to answer that. And I think that in the adults, it's going to be an issue to use gene therapy based in CRISPR. It could be another bottleneck in the future.
Do you have any question, or do you want to share with us what you are doing and what you are looking for in your career, because you are becoming a scientist, right?
Yeah, hopefully.
Okay.
So I'm a student at Texas A&M. I'm going into my senior year, and I'm applying for PhD programs this fall. I want to study essentially delivery of genetic mechanisms, because that's what did a little reading last year when I was picking a lab at Texas A&M, and it seemed like that was like the biggest holdup for applying the genetic technologies that are so exciting that we hear about, is that you can't deliver them. So that's what I want to study in PhD. And I'm applying this fall. I have a little sister who's 6, and she has LCMD, so that's kind of like my motivation for all of it. But I'm here at the conference both to learn how to take care of her better and things like that, and hear about the research, and also because I'm hoping to connect with PIs and people who are doing research in that space, and maybe apply to their labs this fall.
And are you looking in the US or
I think mostly
anywhere?
in the US because of, Kate is 6 and the natural history isn't great, and I want to be around while I can, right? So mostly in the US, yeah.
Good to know.
Yeah. Otherwise I would have probably approached Ignacio and all of you earlier in the conference, but I'm hoping that I can connect with people in the US so that I can stay in the States and be close to my family. Yeah.
And you should do a stage in our labs.
Sorry?
You can do a stage in our labs. You can do a short stage.
Yes. That would be, I would be very interested in, yeah.
So just one thing that we're on sort of delivery and sort of generating these viruses. One thing that we're doing in our lab is, so one of the major problems is that these viruses tend to accumulate in the liver, and that's causing a lot of problems in the clinical trials. And so using the stem cells that we have, we are making from the same patient cells, muscle tissue, but also liver tissue. We can make cardiac tissue. We can make other types of tissue as well. And so kind of one of the approaches our group and other groups are looking at as well is to apply these viral vectors onto these systems in the lab to begin to assess are there specific AAV subtypes that work better in some tissues rather than others, and can we detarget the liver, for example, which is causing a lot of problems. So that's a very active area of research, and I think for you as well would be something you can look at, and there are definitely people in the US who are doing that kind of research as well. So yeah, for sure it's ongoing, and yeah, it's a lot to do.
Yeah, and regarding the vectors, well, maybe you heard about it, and that there is a new generation of AAVs. There is a Mayo AAV that is more, you have to use way less doses of those to get to the same number of cells in the muscle. And most of the research that I'm seeing, people is trying to go to Mayo AAV or AAV Mayo, because there is one in Europe and one in the US, and it still has some problems to overcome, but this is a good solution to reduce the dose, because one of the problems of the AAV is that for muscle that is so big of a tissue, you have to use a huge dose to arrive to all the cells. So people is working on making these capsids that are more precise to target the muscle compared to other organs, and also the AAV evolution. People is using evolution experiments in AAVs to obtain very fast new capsids and test them to see if they go to target and they go to muscle and less to the liver and things like that. So there is also, and gene therapy is huge for many diseases, not only the rare disease. I mean, it's for most of the rare diseases plus other more common diseases. So there is a lot of research going on, and also using other vectors that are not AAV based, like lipid nanoparticles and other things that can carry the genetic material inside the cells without using AAV. So there are some hopes. Still, we cannot say how much time, but it's a huge progress. I don't know if we can wrap up here or more questions.
One more. What?
Yeah.
I guess you guys read about it so much and work on it so much. What have you seen the most effective delivery? Is it like a VLP or one of the lipid nanoparticles, or what do you? If I had to guess, I guess.
Right now, the most effective delivery system is the AAV. That's the one. And probably one of the most, the best right now is the AAV Mayo or the Mayo AAV. But if I had to bet for the future, I think it's going to be some kind of nanoparticle, the one that is going to work. And especially because with the AAV, you only have one shot. Okay, because once you inject the AAV, you are going to be immunized against that one. And that means that you cannot, that's why we want to get a very good efficiency, because we are going to have just one opportunity. But if you have a nanoparticle or something like that, that is no immunogenic, so you can go to the chronic treatment. So you can, even if you have not such a good result in the first shot, you're going to have more chances, and you can accumulate in terms of addition of the DNA. You can accumulate with time. This is not only for the muscle diseases, but also for neuro diseases that you had to go in vivo. I believe that this is going to be the solution at the end, a kind of nanoparticle. I don't know which one, but a kind of nanoparticle.
I broadly agree. I think there is some cool work so one of the problems with AAVs is the size of them, and packaging all of these tools is quite difficult to do because the size of the genes that we need to put into the viruses are basically too large. So there is cool work being done to sort of try and increase the size of the AAV particles, but then, as Ignacio said, there's always the problem of immune response and how you deal with that. So yeah, I think I basically just echo what he said. Yeah.
Plus, there are many other problems that we don't have to think about them right now, but for example, the turnover of the protein, that we know that this protein is quite stable, and if you have the mutant protein there, it's going to take a lot of time to eliminate that protein from the cell, and this is going to be an issue also in the future. And also try to regenerate the tissue because you have a damage already there. How can you revert that damage? That means that you had to also think about in regeneration therapies or something like that. And again, this is not going to be our field also. There are other diseases in which this is an issue, and they are working on that. So that's something that we have to contemplate, to add to our battery of therapies or options that we are going to have to apply to these patients. Because for sure that we have to revert things, and this is not an easy thing to do.
Okay, I think if we don't have any other questions, we can stop here, but we will keep talking during the breaks, and any question, always welcome to receive by email or phone.
Thank you.
Thank you.